Our next speaker is a longtime collaborator of Ilya and myself, Dr. Peter Laird. Um, Peter is a professor in the Department of Epigenetics, and Peter and Imogene Cook Endowed Chair in Epigenetics at the Van Andel Research at the Van Andel Institute. Thank you. And so Peter and Ilya collaborated on numerous projects as part of the Cancer Genome Atlas, including co-leading working groups that focused on and analyzing all the cancers of the GI tract, and worked together on some of the cross-cancer comparisons that you just heard about from Nyasha. And with that, I'll hand this mic over to you, not this one, but this one.
Hello. Yeah. Ilya and I go back, let's see, probably about 15 years or so, 20 years, close to 20 years. But initially in TCJ, he and I just were both sort of new PIs and didn't interact much. And I'll tell you about my very memorable meeting with him. I had the first deep conversation. So today, I'm going to be talking about gastrointestinal cancer genomics and moving on to epigenetics. And in particular, I'd like to highlight the impact of Ilya on my own research. Why is it not advancing? Do I have to? Which one did you? That's what I tried. Okay. So I don't press. So I'm going to start by explaining what DNA methylation is for those of you who don't work with it on a daily basis. Theo already mentioned it. So what is DNA methylation? Well, we about a quarter or so of the bases in our genome, are cytosines. And a DNA methyltransferase can label this with a covalent bond here at the C5 position to put a methyl group instead of a hydrogen to create a 5-methylcytosine. Now, as I'll come back to later, this happens after DNA replication. So it's not built into the genetic code, but it's like a extra layer of information on top of the DNA base sequence. And we have about 28 million sites for potential DNA methylation, which is the CpG in the genome. So that's in a haploid genome. So we have 2 to the power 28 million different permutations of DNA methylation potentially available in the human genome. And then that's just for the haploid genome. So say about 56 million or 2 to the power 56 million, which is a larger number than the number of atoms in the universe or something like that. So amazing coding potential. And the cool thing is, because it's post-transcription or post-replication, each cell can have its own unique DNA methylation code. So what was observed back as early as 1980s was 2 things, and they seem somewhat contradictory. One is that there was a loss of DNA methylation globally in cancer, but at the same time in 86 and a few years around that, multiple reports started to describe focal gain of methylation in cancer compared to normal tissues. So that's shown here. Each of these lollipops here is a CpG dinucleotide. Red is methylated. This open color is unmethylated. And you can see in a normal tissue, you have this low density area of CpGs, which have a lot of methylation, but lose it in cancer. Whereas these clusters called CpG islands, often overlapping promoters, gain methylation in some cases, and this leads to transcriptional silencing, so epigenetic silencing, which can amount to the same thing as a loss of function through mutation or deletion. So epigenetics can act as a inactivator of tumor suppressor genes, for example. So I want to now move on to how I met Ilya for the first time for real. So we'd been introduced to each other at steering committee meetings and of the TCGA since probably 2008 or so, 2007. But it was at this meeting in 2012 on the French Riviera. It was actually an ICGC meeting where at the opening reception, I was sort of milling about, not knowing all these ICGC people, Ilya doing the same thing, and we bumped into each other and started chatting. And it was first of all, it was a very memorable conversation. I remember it. It's 12 years later. And it was one of these situations where there were two people who were very keen, very curious and keen in getting to understand the other person, but we were speaking different languages. So Ilya was an engineer using math and talking about Boolean networks and things like that. And I was trying to explain to Ilya what DNA methylation was. And back then, he didn't really know exactly what it was or how it worked. And so with these two people, we struggled for maybe 10 minutes or so, and then we started to find common ground. And ultimately, it led to something that influenced my research for quite a while after that. And that is the work of Theo with this paper that came out 2 years after that first conversation. And we started talking about how to visualize both this focal gain of methylation and this global loss of methylation at the same time. And he said, well, this is something that we've been working on. And so we ended up collaborating with Ilya, and in particular, Ben Berman in my group worked with Theo to create this figure where you show here in a log2 scale horizontally the sort of focal gain of methylation in cancer and the global loss on the same image in the same representation. So that was really cool. This was the DNA methylation figure in the paper, but we actually found this, this representation over here where the scale is on the vertical axis and the chromosomal position here on the horizontal axis to be more useful and helpful in the long run. And so that's what we've been using since then. And this just shows you a paper from 2018 that we published on the linking DNA methylation loss to replication. And I just put this up here because 5 out of our 8 figures use some form of multi-scale representation. So if you look there, there are all these different variants. These are all multi-scale representations, and I'll go into, and even down here. And sometimes it was for DNA methylation. We even showed multi-scale representation of cross-sample standard deviation, for example. So this was 4 years after the multi-scale paper was published, but it still continued, and to this day is a very useful way to visualize DNA methylation changes. I want to highlight one of the examples because it's so cool to visualize here. The changes that happen during aging, you can see very nice with this multi-scale representation. Here in yellow is high DNA methylation, blue is low. This is the whole genome of a newborn CD4 T cell. And here are the scales from 10 megabases down to 10 kb. So fairly uniformly methylated, with some of these CPG islands barely visible because of the scale. We're looking here at chromosome 16. And if you look at the same cell type, but now in a 103-year-old, you can see these gaps that have appeared where there's been loss of DNA methylation, but it's not uniform. It's in these particular locations. And if you look at cancer, this is a T-cell prolymphocytic leukemia from the same cell type. You see that it's even more exacerbated here. And so we asked ourselves, what is so special about these regions? And we found that they were associated with lamina attachment, which means DNA that is at the periphery of the nucleus, so around the outside, which tends to be gene poor and late replicating as well, which you see here. These had been identified by Joe Ecker's group as partially methylated domains. And here we make this link to show that there's a relationship with age. And why would that be? Well, let me explain how DNA methylation gets onto the DNA. I mentioned it earlier. If this is a methylated CpG, it's palindromic, so there's information on both strands. And after DNA replication, that information is conferred to both daughter cells or daughter DNA molecules. The newly synthesized DNA is unmethylated and gets remethylated by the DNMT1 DNA methyltransferase to restore the pattern if there's a methyl C in the parent. If there's not, it doesn't. But what happens if that process fails? What you end up with is a retained hemimethylation state. If you then go through another round of DNA replication, you end up with complete loss of methylation. And since this is a maintenance methyltransferase, it's very difficult to get that back again once you lose it. So what that means is we have a process in our bodies that copies the DNA methylation state very faithfully, 99% plus accuracy with every single cell division. But if you do 99% or 0.995 to the power, whatever number of cell divisions, you end up slowly losing DNA methylation over time with each replication. So the question is then, can you use that as a replication clock, as it were? And we showed that you can, and we published this 2 years ago. We identified a sequence that is particularly sensitive, and that is a CG surrounded by an A or T in a CpG poor region. This loses it the fastest. It's often prevalent in these PMDs, these late replicating regions. And in various different cell types, when we measured, this is cultured in vitro with population doublings out to about 100 or so. Every single cell type that we looked at had this uniform loss of DNA methylation at these solo WCGWs. So we went on then to use this to develop a clock that measures replicative history, which is cool because if you had a pure cell population, you can now say, how many cell divisions has this gone through? The challenge for us now is if you take a primary sample, like a blood sample, is to do simultaneous deconvolution of cell composition and this replication clock. And so that's work that's ongoing. So now moving on to the TCGA era. This is 2013 in front of ISB. You'll recognize that. Ilya here and the co-chairs of the stomach cancer working group were Adam Bass, Ilya, and myself. And I think Vestin is in here he's hiding in the back there. That's Vestin. And so we had a great time and made a lot of advance. And we came up with this way of a new classification of gastric cancer, stomach cancer, where we divided into 4 molecular subtypes, including one associated with Epstein-Barr virus infection. And this led then, it was part of this paper that has been mentioned, or I think already today, there have been so many TCGA papers mentioned. I wanted to point out, this paper has been cited as of this morning more than 5,000 times and has had a real impact in the clinic. And so I wanted to point this out as one of the lasting legacies of Ilya's leadership, that this has had a major impact. It's one of the most highly cited papers in TCGA that Ilya helped to lead. So this is one of the things that will live on long after all of us probably are no longer here. This is the image that we had in the paper of a stomach with the different subtypes and where they are and some of their features. And this image was, if you Googled gastric cancer classification, this is what I did this morning, you'll see that it pops up here. It used to be near the top, but what happened is all kinds of other people started to copy this, and now they come up higher, which is fine. But it shows you how this work that Ilya helped to lead has been disseminated and absorbed into the larger clinical culture. So 2 years later, we moved on up the GI tract a little bit to the esophagus, and many of the same people. Let's see, where's everyone? There's Vestin, there's Ilya, and there's Adam. And this was in Boston at the Broad Institute. And I love this picture. Ilya, Adam and myself and Vestin. And Vestin is, I should mention that he's really was instrumental throughout all of these projects, really very much driving them. And this paper also has had a big impact, 1,300 citations so far. This is the image that we had. And one of the main conclusions was we did sort of an integrated analysis of both gastric cancer and esophageal cancer and found that there was no sharp border. And actually, surgeons, when they resect tumors near the gastroesophageal junction, they call it one or the other, but often they're growing over the junction, and it's not always easy to tell where they're from. And because of reflux and Barrett's esophagus, you often actually have very similar molecular features in the lower esophagus tumors as in the proximal stomach. So it's more of a gradient, an important result. Then we moved on to include the lower GI too. And so this is now entire gastrointestinal adenocarcinomas, focusing on adenocarcinomas and leaving out the squamous cancers of the upper esophagus. And the reason I'm showing this is you'll notice that Ilya is not mentioned here. And I felt that it was important to mention that part of Ilya's mentoring and generosity was making sure to promote and help and advance the careers of people working in his lab. And Vestin was now one of the co-chairs and did an outstanding job. Ilya working behind the scenes, continuing to help us, but Vestin very much a driver in this project. So now I'd like to move on to how we've been advancing cancer epigenetics after this TCGA period. And I wanted to point out first this EBV, Epstein-Barr virus-associated stomach cancer I mentioned earlier. That turned out to have extremely high levels of this focal promoter DNA methylation that I had been talking about. The highest is the champion really here, with about 20% of its promoters methylated, which was quite unusual. And there's been some work since this paper came out on mechanisms, induction by viral proteins, for example. And so in the rest of my talk, I'm going to focus a little bit more on this focal gain of methylation. And several groups, ours was one, and then Steve Balin and Howard Cedar's groups published in the same issue of Nature Genetics back in 2007, that if you look in stem cells, there's this complex called polycomb repressive complex, which occupies certain promoters, about 1,000 or 2,000 or so in the genome, and represses them transiently. And the promoters drive master regulators of differentiation and development. And so these are kept poised, ready to be turned on as the cell needs to differentiate. They're in a poised state. So the model that we came up with, so the question is, what is special about marking these gene promoters in stem cells, and why do they become DNA methylated in cancer? What is the link between them? And the model that we proposed is that you have this adult stem cell with PRC, polycomb repressive complex, at this master transcription factor promoter. And upon differentiation, you get this master transcription factor turning on, and it drives all these lineage-specific genes, and the cell starts to differentiate. Well, what happens if there's a loss of polycomb repressive complex and DNA methylation steps in? DNA methylation is associated with a much more heterochromatic structure and is a much more permanent way of silencing gene expression than this transient state here that can easily be released. And so if you get this block here of expression by DNA methylation, then this transcription factor can no longer turn on. And so essentially, you have, at least depending on the transcription factor, a partial block in differentiation in this stem cell, which is not a cancer cell. It's just sitting here, but then but it has this block. If it now gets a strong cancer driver mutation, it can develop into a cancer cell. And we believe that is why when you look at the fingerprint of where methylation is in cancer cells, it's reflective of where the polycomb was in the original stem cell. So we're working hard to sort of validate this model, and we found evidence for these stem cells, for example, in mice with LGR5 cell sorting. We can see evidence for this polycomb target methylation. But we're also doing this in primary human cancers as well. And this is a project that is ongoing. It's unpublished, but it's a big project working with local hospitals to collect colorectal adenocarcinomas. We came up with this custom designed embedding system where we then slice the tumors into 3-millimeter sections. These are then mapped and biopsies taken that are spatially tracked. And then there's a whole bunch of different things that we do. I won't go into all of them, but it includes infinium arrays, but also single cell whole genome bisulfite sequencing to look at DNA methylation. And I wanted to show you some of the interesting results. And one of the things we found is this is a single case where we had 63 different locations in the primary tumor. And this is looking at DNA methylation clusters, clones, subclones. And you can see that clearly there are two main subclones. Red is highly methylated, and each of these rows here is a different location in the genome, and blue is unmethylated. So this one has more methylation. This one has gone down a different phylogenetic branch. So when we then looked at where these different samples mapped in the primary tumor, we can see that they're spatially separated. And here's the tree up here, and you can see how the spatial location maps to the original phylogeny. So we're continuing to advance this and making monoallelic versus biallelic calls. But I wanted to show you a cool result where we took a slice and did cyclic immunofluorescence. And Toshi Hinaway in our lab looked at the two different clones and identified differential DNA methylation between them, one at SAT2 promoter and the other one at CHP2. And these are genes that are normally expressed in the healthy normal colon. And so the prediction would be that if you had promoter methylation, you would have if you had CHP2 promoter methylation, so this purple one, you would have low expression, which would be in subclone 2, and vice versa for SAPV2. So then we ordered antibodies to these proteins and took this middle slice, and this is just showing so we did cyclic immunofluorescence with a whole bunch of different markers, but you can sort of see the structure here. And the beta-catenin is in this aqua color, so you can see where the tumor is. And then when we looked at the two clone-specific antibodies, we looked at these, we profiled these two biopsies. One was from the subclone 2, which is methylation of CHP2, so it should be green up here. And this one should be more of this magenta color down here. And that is, oh, sorry, I mixed it up, sorry, the other way around. So this one has methylation of the green one, so that's why it's pink up here or magenta. So this shows how we can actually use subclone information to then go at very high resolution, see exactly where the subclones are. And we can do that now for the whole tumor and map how these are spatially represented, and then compare that to the phylogeny and try to understand epigenetic evolution of primary human cancer. Now, finally, I'd like to go into single-cell epigenetics. You've probably seen many papers on single-cell TAC-seq, for example. Single-cell DNA methylation is not easy, and it typically get very low coverage, like single base pair, single percent coverage, 5% or so. So we wanted to improve that. And why did we want to do that? Well, if you look at if you're trying to detect these rogue cells that may have a lot of methylation, you just do bulk sequencing, you get essentially a mishmash here of everything mixed up. But if you have a cell here that has a lot of this polycomb target hypermethylation, and you take a bulk sample, you would get the same result. So you can't distinguish between these two scenarios. So for this reason, we've developed this new, we call it SCDMC method. It's on bioRxiv now. We show that we can get 30 to 40% coverage. We've actually gone a little further than here with high sequencing efficiency, and allows us to actually identify for every single cell what the cell type is. And here you just see primary or epithelial mucosa, where we can separate lymphocytes from intestinal epithelial cells based upon loss of methylation of cell type specific, mostly enhancers, but these are cell type specific loci. So we've been using that now to look at some of these primary human cancers. And this is showing you one where we had used the whole genome bisulfite sequence data to look at copy number alterations. And so you can see, for example, loss of chromosome 11q and chromosome 14, et cetera. And we did EpCAM sorting, and you can see that in this particular biopsy, most of the cells were either normal intestinal epithelium or EpCAM negative, and only a small set here had these copy number alterations. And then when we looked at the solo WCGW methylation, which is an indication of how many cell divisions a cell has gone through, the cells with, this is a single EpCAM positive cell with copy number alteration. You see more of a yellow-green color with these gaps here, and that indicates loss of methylation, whereas a EpCAM positive cell without copy number alteration, so probably a normal epithelial cell, has much higher. So this cell here from the cancer has undergone far greater number of cell divisions. So this is cool because that means for every single cell, we can actually assess how many cell divisions the cell has gone through. And here, I'm just going to end by showing you some NMF. These are the 3 NMF factors here, looking at solo WCGW. And we had really sort of separate groups here. And this is showing, so the darker the color, the more cell divisions the cells have gone through. So there's this group up here and this group down here. What else can we learn about these? Well, if we look at the percent of CpG islands, so these polycomb targets, for example, that are hypermethylated, we see up here a gain of CpG, this focal methylation, whereas this other group, which also had a lot of cell divisions, does not have as many of these CpG islands methylated. If we now look at enhancers, red indicates low methylation at immune cells, meaning they're active in immune cells. And lo and behold, this group down here, that is some of which have undergone a lot of replication and cell divisions, are actually immune cells, whereas blue is low DNA methylation in colon epithelial cis-regulatory elements, so active in colon cells. And so these are the colon cells. These have the CpG island hypermethylation, these don't. So if we now look at which ones have the copy number alterations, that's this group up here. So we can see immune cells, normal colonic epithelium, and cancer cells. This was a first pilot experiment. We're doing this on a larger scale now. So take-home messages are this widespread hypomethylation is tightly linked to cell division and can be used as a mitotic clock. We call it Replitally. Polycomb target CpG islands hypermethylation starts in normal tissues and continues during tumorigenesis and may indicate cancer risk, we think. And Ilya has had a lasting impact on my research and has left a legacy with clinical implications in gastrointestinal cancer. Just very briefly, two of the people I'd like to point out, Toshi, many of you have been on calls with him here who've been in the TCGA project. He's been a longstanding DNA methylation analyst in my group. And Nathan Spix is leading the charge for this single cell and primary human colorectal cancer work. And I'd like to end with one of my favorite photos of Ilya. Not because he's so materialistic, but this shows another side of Ilya in terms of leadership. This was at the ISB meeting in 2013, and here he's collecting money from everyone to pay the bill. But the reason I like the photo is just the sparkle in his eye and just the warm, friendly aura that was always surrounding Ilya. So thank you, everyone.